Alright, in this video we're going to look at cell signaling. So, how cells communicate. And that can be how they communicate with each other, or cell communication, meaning how a cell responds to stimuli. So something that interacts with the cell, or something that's happening the inside or the outside, how it responds to different situations, to different stimuli. And my picture for today is a kind of a schematic or a diagram view of all of these messengers proteins in the cell that can pass along the message that of when a stimulus binds to one of these receptors on the plasma membrane, and it can trigger a cascade of different messenger proteins, taking this signal and relaying it to different places like the ER, the nucleus, or different locations in the cell where a response might happen. So there are hundreds and hundreds of these different relay proteins, these messenger proteins as they're called, that help take the message from wherever it's received to where the cell can do something about it. So cells need to communicate with each other so that, like, all of the cells, say in your hand, can, like, all the muscle cells can contract in a similar manner so that you can hold a pencil, for example. So they need to communicate with each other, but they also need to respond to a stimulus. So what might be a stimulus? Say like a pathogen or a bacterial cell, that's next to them, or a virus, or different conditions, changes of acidity, ions, different things around it that it may need to respond to. It has to send signals either to other cells or into itself to trigger different things that needs to happen. Cells can communicate locally, so when they're near each other. And some examples of signaling between cells that are near each other is like synaptic signaling. So your neurons, they have these neurotransmitters, which are messengers or signaling molecules that excite or pass along the electrochemical signal between neurons so they can communicate and send information all through your nervous system. You also have endocrine signaling for cells that are really far away from each other. So, for example, your pituitary gland secretes a lot of different endocrine hormones that can go and affect or tell other cells to do something very, very, very far away in your body. Like testes in males respond to testosterone signaling that's produced or helped to be produced in your endocrine glands in your brain. So sometimes these signals can go over vast distances in your body, like hormones produced in your pituitary gland can reach all sorts of different target cells in your body. Another example being insulin in your pancreas that can have an effect on all the cells in your body to tell them to start bringing glucose in. Some ways that cells that are really close to each other, so adjacent to each other, communicate is via direct contact. So like this picture, maybe these little balls represent glycoproteins or glycolipids that stick out of the extracellular matrix of one cell, and they might fit into these little receptors in a different cell next to it that tells the cell to do something. So in cell-to-cell recognition, signaling is important. Also, molecules can flow through like the gap junctions in animal cells and the plasmodesmata in plants. Remember, those are those little holes kind of connecting cell walls in plants or membranes in animals. And chemicals dissolved like in a cytosol might flow between the cells and signal the cell to do something specific. And it just goes through those holes, those gap junctions, or the plasmodesmata. And the last thing is membrane-bound receptors might help in the signaling pathway. So, for example, if you have a pathogen that might bind to a specific receptor cell or receptor protein on the plasma membrane of like a animal cell, once that receptor protein receives the signal from the pathogen, it might trigger this cell. Maybe this is a cytotoxic T cell to kill this pathogen. So, for example, here's kind of a review picture about local signaling. So secreting specific chemicals or different signaling molecules to alert other cells to a change in condition. Long-distance signaling, like hormones in the endocrine system that might travel through the blood, and synaptic signaling between neurons that release those neurotransmitters that are the signaling molecules. So cell signaling is kind of a process that proceeds in 3 steps. So in the first step, a cell receives some sort of signal. Any of those signals we talked about in the last page, hormones, plasma membrane bound receptors, and physical touch of 2 different cells, it might receive that stimulus, and there might be a receptor protein, or maybe just the plasma membrane physically feeling another cell touch it, might be the reception. So the reception is kind of the point of contact where the signal that's supposed to tell a cell to do something and the receptor, usually a protein, meet. They come together kind of like a lock, the receptor and the key. And when the receptor receives that signal, it then triggers the cell to pass that message along to whatever part of the cell can carry out the function that's associated with that signal. The second part, transduction, is just passing that signal or that message along that was received towards wherever area of the cell or components in the cell that are capable of making a response to that signal. The third part is the response. So depending on the stimulus or the signal, the cell may need to do a wide variety of things. For example, there might be a signal to tell the cell that it needs to start producing more protein, or take in glucose from outside, from the bloodstream, like if the signaling molecule was insulin. The response that the cell does after the signal is received in that correct location. So the response is basically what happens, what does the cell need to do when it receives this signal. Right. So reception. Reception in your cells is largely mediated by things called receptor proteins. And these are integral proteins, membrane-bound proteins, that have a little spot in them, or a little site where signaling molecules from other cells or the same cell can bind into, and that receptor protein will then change. Well, its conformation will change, or it might become dislodged from the plasma membrane, and it'll start to pass that signal along. So some receptors on the plasma membrane and some are inside the cell for signaling molecules that can get through the plasma membrane. So not all of these receptor proteins are on the plasma membrane. Some might be in the cell on the nuclear membrane, or some might be attached to different membrane-bound organelles to receive signals from molecules that can make it through the plasma membrane. G-protein coupled receptors are a class of these signaling or receptor proteins on the plasma membrane that are really important. And over 60% of medications work by altering this G-protein receptor pathway. So, for example, if you have an antihypertensive medicine, for example, those work by interrupting the communication in this G-protein coupled receptor pathway that tells your cardiac cells to pump harder, or stopping your cells in your blood vessel from constricting. So G-protein coupled receptors are called that because they incorporate something that's called a G-protein. And kind of in this picture, this is the G-protein, this is the receptor. So a signaling molecule would come in here, and the G-protein becomes activated when GTP, guanosine triphosphate, it's another energy molecule related to ATP, when GTP is used to activate this molecule. So for example, in a G-protein coupled receptor, a signaling molecule might bind to the G-protein coupled receptor, and it will then signal to this G-protein that, hey, we've received a signal. GTP will be used to kind of activate that G-protein, that then passes the message on to this activation enzyme, which is responsible for kind of signaling the cell or telling the cell what to do. So G-proteins are really important. They use the power of GTP to send the signal along in your cells. There's also things called ligand-gated ion channels, and a ligand is just something that binds onto the protein. So for example, in the G-protein coupled receptor, like we said before, this signaling molecule would be the ligand. Kind of what is the key that goes into that site and tells it to do something. So ligand-gated ion channels regulate the entrance and the exit of ions using that key and lock kind of model. So, for example, this ligand, this molecule, might bind to this closed gate channel protein, and when it binds onto this gate protein, this channel protein opens and lets these ions in or out. So that's an example of a ligand-gated ion channel. And receptor proteins inside the cell often turn on and off transcription of DNA. So ultimately what most of these signals end up telling the cell to do is to kind of pass a message along, usually ultimately into the nucleus, and tell it to start or stop transcription of specific proteins. And usually it does that by signaling the molecule that ultimately goes into the plasma membrane to be a transcription factor, or a helper to start making and transcribing certain proteins. The second step is transduction, and that's really how a signal is passed along from reception to response. And in any case, what happens is there's usually a cascade of different secondary messengers. So those are those things that pass the signal along. So, for example, in our G protein linked receptor, this CAMP, CAMP, and protein kinase, and all of these things are secondary messengers. One activates the other, activates the other, activates the other in a long chain until ultimately you get to the point where something is responding. So maybe 5 or 6 secondary messengers down in this chain, a protein would be activated to be a transcription factor, to transcribe some DNA to a protein in the cell that serves a specific function. So all these intermediaries are kind of the secondary messengers. CAMP, or cyclic adenosine monophosphate, is an important secondary messenger. For example, in G protein coupled cell signaling, the messenger kind of binds with the G protein linked receptor, which with GTP activates G protein, which binds with this molecule called adenylyl cyclase, which then through ATP activates cyclic adenosine monophosphate, which activates protein kinase A, which down the line until there's a cellular response. Another important way that a signal is transduced is by the phosphorylation of intermediate molecules called protein kinases. So, for example, a signal, maybe this is epinephrine, binds to a receptor protein, which then activates this relay molecule. This relay molecule activates protein kinase 1 into an active protein kinase, which then activates a different protein kinase, which then in its active form activates a third, which then activates a protein that does a certain cellular response. So in this chain, we call it a phosphorylation cascade. What's a protein kinase? Well, a protein kinase phosphorylates or adds a phosphorus onto a molecule. That's what a kinase is. It adds a phosphate group onto a molecule. It phosphorylates it. So this is called a phosphorylation cascade because protein kinase 1, when it's activated by this activation molecule, phosphorylates or adds a phosphorus group onto protein kinase 2, which then phosphorylates protein kinase 3, which then phosphorylates this inactive protein, which can then do a cellular response. So this phosphorylation cascade, phosphorylation really is just activating. They're activating one another, and that activation is passing the signal along until it actually can do something. You have these little purple things called protein phosphatases, and what they do is dephosphorylate molecules. So after the signal has been passed along, these protein phosphatases come in and unactivate protein kinase 3, 2, and 1, so they kind of reset the system so it can receive another signal. So these secondary messengers, like in the phosphorylation cascade and the G protein linked reception, become activated by others. Usually that activation is through being phosphorylated or through ATP or GTP energizing the molecule. And finally, there's a response. So at the end, a response regulates some activity in a cell. Many responses ultimately regulate the synthesis of a protein, like we said. After the phosphorylation cascade, this last protein might come into the nucleus and serve as a transcription factor for a specific gene to make a specific protein that the cell might need. Other times, the response is the activation or the changing a protein or cell certain cellular machinery. So, for example, if what you call insulin binds to a receptor, the response might be a channel opening up in the plasma membrane, like 4 or 2 receptors that take in glucose. So not always is it a protein being synthesized, but a lot of times it is. Responses may happen anywhere in the cell, so on the surface, in the nucleus, in the cytoplasm. Some things that are responses to a signal might be telling the cell to divide, or to excrete other cell signals or chemicals to tell other cells something, to create a new cellular protein, to bring in certain molecules from the outside. All of those are examples of responses. And this is basically how the cell can respond to internal and external changes in its environment. All right, that should be it. If you have any questions, just let me know. Otherwise, see you in class.